Okay, hello everybody. I'm Sagiv Shifman from the Hebrew University. As you heard, I'm going to talk about the relationship between genotype and phenotype in the context of neurodevelopmental disorders. And what I would like to emphasize in this talk is that we can use functional genomics, so not just reading the DNA, but also manipulating the DNA to better understand mechanisms, predict new risk genes, also use it to predict phenotypes. So I'm originally was focusing on autism as the neurodevelopmental disorder that we're interested in. So this is one of the reasons is because it's very common in the population. It's a major challenge. The global estimate prevalence of autism, this is true for 2021, was 1 in 127 individuals. This translates to around 62 million people in the world, and so really a big issue. But we do know that there is a very strong genetic basis for autism, and there was a huge, tremendous success in the last years to identify also the specific genes that are involved in autism. So, for example, in this study published 3 years ago, they were able to identify 72 high-confidence genes for autism and another set of 309 genes that are high-confidence genes for more broad developmental disorders, and the two sets are overlapping. Here are the 72 genes. Most of the evidence for those genes are coming from, first of all, de novo mutations, from new mutations, and most of the evidence also comes from protein truncating variants. Okay? Now, still, so those are genes that are associated with autism. I already told you that they're also more globally associated with neurodevelopmental disorders. And there is quite a variety of phenotypes that are associated with each one of those genes. To emphasize it, I want to look at one particular gene, POXZ, which I like very much because we extensively study it in my lab. So people with mutations in POXZ have a variety of phenotypes, some of them with very complete penetrance, like developmental delay and speech delay, but there is a very large list of symptoms and phenotypes that with incomplete penetrance, and this includes microcephaly and also autism. So we have big challenges in autism research that includes the fact that most individuals with ASD diagnosis lack those rare and de novo variants in high throughput, in high confidence genes. So we need to develop new methods to identify the genes that are involved also in the more complex inheritance. Another challenge that I've just showed you is this diagnosis and phenotypic diversity that we see even for individuals with very similar type of mutations in the same gene. But there are genes that are more likely to result in a ASD autism diagnosis versus others that are more likely to lead to other developmental disorders. And you also saw that there are many genes involved. They also involved in diverse molecular functions, and one of the questions is, do they converge on specific processes? So one hypothesis that you can see in the literature is that autism genes converge on their role in proliferation and differentiation of neuronal progenitor cells. So you have different molecular pathways, but they all affect this balance between proliferation and differentiation, which could explain why you see some kids with also changes in their brain size, including macrocephaly and microcephaly in autism. So to test this kind of a hypothesis in an unbiased way, Alana Amiran, a PhD student in my lab, she designed a screen for genes that are essential for neural differentiation. And the way she did that was to take mouse embryonic stem cells, transfect them with guide RNAs that target almost all the genes in the genome in a way that in each cell you have a mutation in a different gene. And then she took those cells, split them into two groups. One group went through differentiation to neural progenitor cells and neurons. The other group kept as embryonic stem cells. And by comparing the two screens, she could identify those changes that are specific to the neural differentiation. And by this, she identified 331 essential and 110 growth restriction genes. It means essential genes means that their guide RNAs deplete over time. It means that the loss of this gene reduces survival or proliferation of the cells. And there is those growth restricting genes that you see the opposite. The guide RNA enrich over time. And here are two examples of genes known to be associated with neurodevelopmental disorders. In red, it's ADNP. You see that during the differentiation, there is a reduction in the guide RNAs readout. And for P10, we see the opposite, a known also gene for neurodevelopmental disorders, but also for cancer. We see regulation, or it's not upregulation, it's actually increase in the amount of guide RNAs that target this gene. You see the similar trend in embryonic stem cells, but this is not significant. So we've went to study some of those genes in the screen, both in vitro and in vivo. In vitro, what we did, we used a SOX1 GFP system. SOX1 is a marker for neural progenitor cells, so we can use it to quantify the differentiation efficiency. And for those 4 genes that we've studied, that we took randomly from the top hits, we see for all of them reduction in differentiation efficiency and increased cell death. We also wanted to know what's going on in vivo and looked at the structure of the brain for a knockout of 8 other genes. This is work we did with Binaz Yalcin and Stefan Collins. We saw in all the cases that the knockout of those 8 neural differentiation essential genes cause severe neuroanatomical defects. For 4 of them, there were specific defects that we saw. And for another 4, which I'm going to show you now, we see microcephaly. So the brain in general is smaller. This is what you see here. On the y-axis, it's the percentage from the wild type for different neuroanatomical features. The color represents the significance. You can see in general that all those bars go down because there is reduction in size. But still, there are some regions that are more significant than others. For example, for PEDS1, in addition to the smaller total brain area, we see that the internal capsule is much smaller. This is an important structure, a white matter structure that connects the cerebral cortex with other subcortical structures. So now how this relates to human brain phenotypes and disorders. We did general enrichment analysis, and we found that not only that they are associated, but the stage at which the genes are essential can explain some specific disorders. We looked at 4 different groups of genes here, and those genes that are essential before differentiation in the embryonic stem cells, those that are essential in different time points during the differentiation, and the growth restricting genes, and we compare it to a list of genes known to cause or be associated with microcephaly, macrocephaly, loss of function intolerant genes, genes associated with developmental disorders or autism. This is from the same study I've showed you, but with a less stringent threshold. And this is the enrichment. I want to focus here on one thing which is, I think, very interesting, is the differences between developmental disorders and autism. So for developmental disorders, you see there is a significant enrichment for genes that are essential even before differentiation and with the growth restricting genes. But for autism, we see the opposite, a mirror image. They are enriched only with the genes that are essential during neural differentiation. Now, while there is this enrichment of neurodevelopmental disorders with the genes that we identified in the screen, most of the genes we identified, around 80% of them, are currently not linked to disorders. And so we wanted to see if we can use it as a resource to identify new genes. We focused on the 8 genes that also show neuroanatomical changes in mice. And for 5 of them, we could find patients with mutations. In most cases, they had microcephaly as well. But in the end, we decided to focus on just one of them, which is PEDS1. And we show that pathogenic variants in PEDS1 cause a novel, undescribed autosomal recessive disorder. We did it with work with Nadira Damesh and Tamar Arel. So we've managed to identify 2 families these are families with consanguinity, that their inheritance patterns match a recessive inheritance. The PEDS1 is part of a known pathway of plasmalogenes biosynthesis. All the genes that I'm showing you here from this pathway are known to cause also neurodevelopmental disorders, and we see shared phenotypes that includes developmental delay, microcephaly, congenital cataract spasticity, and seizures. But very interestingly, PEDS1 and FAR1 deficiency lack some of the skeletal abnormalities that are seen for this group that I've just circled of genes that are all function in the peroxisome and belong to this RCDP syndrome. And it's possible that where those proteins are explain the differences in phenotypes. So now we went to look at the function of PEDS1 in brain development, again looking at in vitro and in vivo systems. First of all, we've generated mutations in PEDS1 in mouse embryonic stem cells to look at how they differentiate, again with the SOX1 GFP system. And here we see an opposite thing with what I've showed you for the other 4 genes, that actually the differentiation efficiency goes up. So we see more cells with SOX1 GFP. But when we do RNA sequencing on those cells, it looks like the differentiation itself it's more complicated than just accelerated differentiation. It looks abnormal. We see hundreds of genes that are differentially expressed. This is the pathway enrichment analysis. We see downregulation of nervous system development, for example, and upregulation of genes that are involved in pluripotency, mesodermal commitment, some activation of Hox genes, but also upregulation of genes that are involved in cholesterol and fatty acid metabolism, which makes sense. We then went to look at the effect of PEDS1 in vivo. This is work we did with Koichi Nagata and Anako Amada. And we see that PEDS1 deficiency impairs both the migration and proliferation during cortex development. The technique we used is in utero electroporation. You electroporate DNA construct into subpopulation of progenitor cells in the ventricular zone of embryonic brain, and those progenitor cells, of course, will carry the DNA and they will differentiate, migrate, etc. We've first looked at the cell cycle exit. This is done by injecting both construct with GFP and shRNA, knocking down PEDS1, and then after 24 hours you inject EDU to label the proliferating cells, and after another 24 hours the brains are dissected and are labeled with Ki67, and so we compare those cells that were proliferating and are still proliferating, labeled still with Ki67, and those that exit the cell cycle and don't have the label of Ki67, and we compare controls to shRNA against PEDS1, and we see an effect, a significant effect, meaning that the cells with knockdown of PEDS1 exit the cell cycle much faster, meaning they are probably differentiating faster, which is similar to what we saw in vitro. We also see a very clear, striking migration defect. Here you see the control cells that they migrated to the upper layer of the cortical plate. This is on the left. And you see different shRNAs that, with different efficiency, that knock down the PEDS1 gene and affect the migration of the neurons. Number one, with the most defects, also have the strongest effect on knocking down the protein, and we can also rescue partially the phenotype by re-expressing a PEDS1 which is resistant to the knockdown. So now I want to move to a more focused type of study that we're doing by another PhD student, where he focuses on many autism genes, and he couples the CRISPR screen with also single-cell RNA sequencing, and he does it in 2 stages during differentiation to cortical neuron. And this is Elad, is the PhD student. He is co-supervised also with Eran Meshoel. So we've designed a guide RNA library to target 39 high-confidence autism genes. We have also 32 non-targeting control guide RNAs. They were transfected into human embryonic stem cells, of course, expressing Cas9. And we look at 2 time points after 7 days and after 21 days of differentiation. I didn't mention it, but these 39 genes were all the high-confidence autism genes that are involved in transcription and chromatin regulation. And so single-cell RNA sequencing reveals a variety of cells, including progenitors and neural stem cells in different stages of the cell cycle. Those are mainly identified on day 7, here in orange, and also more differentiated cells that we identify mainly after 21 days of differentiation. And we can also reconstruct the differentiation, and this is it. I mean, most of the cells, they originate from the neural stem cells, and then they differentiate into these 4 trajectories leading to neurons, ependymal-like cells, oligodendrocyte cells, and also VLMCs, that stands for vascular leptomeningeal cells. So this is all the cells that we're observing. And of course, the question is how the perturbation of autism genes affect those cell composition. And so what we did was to use randomization, downsampling, to compare the perturbed cells with the control cells, because we have much less control cells. And this shows that the perturbation in general of autism genes promotes differentiation to neurons but reduces the VLMCs. And this is just the result showing this significant effect for both cell types. But this is over all the autism genes, but we do see specific effects for different genes. Here is an example. You can look at, for example, KDM5B goes with the trend of increased neuronal cells and decrease in VLMCs, but other genes that I've mentioned, like ADNP, we see actually increase in oligodendrocyte cells. So we can use these cell composition changes to cluster the autism genes into different groups. Here are the four, we have actually 5 groups, but one group is just one gene alone crying here by himself. But we have those 4 groups, but quite remarkably, those 4 groups are associated with brain phenotypes. So I'm showing you here whether the genes are involved in microcephaly and macrocephaly. Red is true here. And you see it doesn't distribute randomly here. We have 2 clusters, 1 and 4, which are more associated with microcephaly. In both of those clusters, we see more oligodendrocyte-like cells. Another cluster here, number 3, which I don't know if you can see it because our small colors here, but number 3, we see increase in the number of neurons. This is more associated with microcephaly. We next wanted to see if we can use the data from the screen to predict whether a gene will be more likely to cause autism than neurodevelopmental disorders. And I'd use a method called linear discriminant analysis to distinguish between those ASD and DD predominant genes. This method looks a bit like PCA because of the reduction in the dimension, but unlike PCA, we are trying actually to maximize the differences between the groups. We have 3 groups here. Autism predominant genes are in red, developmental disorder genes are in blue, and gray are genes that are in between. And we have the features here also that contribute to these separations. This is some of the features, including for example, frequency of VLMCs and frequencies of quiescent neural stem cells are contribute to the separation. But also, I want to emphasize that when the genes are expressed, whether they are highly expressed after 21 days or whether they are expressed after 7 days, is also contributes to this separation in a way that genes that are expressed earlier are more likely to lead to developmental disorders rather than autism. And we validated this also by looking at another data set of bulk RNA sequencing during the same procedure of differentiation to cortical neurons. We separate the genes into 2 groups. Red is higher expression. So we have the group here which genes are expressed during the first 12 days in a higher level, and the other group which are expressed later in the differentiation. And we compare it to the distribution of the posterior probability for ASD diagnosis. The posterior probability means that if it's closer to 1, more likely we'll get an autism diagnosis. Here you see that there is, for the early expressed genes, higher probability to get a developmental disorder diagnosis, while for late expressed genes, we see much more likely to get an autism diagnosis. So this validates what I just showed you before. So in conclusion, our study provides a genetic platform to identify new NDD genes, also predict the likely inheritance patterns. I didn't show you that, but based on the pathways that are involved, we can predict quite accurately whether the inheritance will be recessive or dominant. We also found that ASD gene perturbations accelerate differentiation towards different lineages that result in cell composition, which is associated with human phenotypes. So thank you very much for listening. I thank my lab members, which are listed here, our collaborators, which I've mentioned along the way, and our funding agencies. And thank you very much.